how nutrients are recycled in our ecosystem to provide us with the energy we need for our daily activities. Ever wonder what happens to a leaf after it falls to the ground?
First, the leaves fall to the ground. Then they soak up water and soluble compounds are released. Along comes an insect or worm to start breaking them into smaller pieces. Now that they are in very small pieces, the microorganisms can begin their work.
Internal cycling or nutrient cycling is an essential feature of an ecosystem. It is the process where the microbial decomposers transform organic nutrients to mineral form ready to be taken up by plants. Nitrogen is an essential element in nutrient cycling. Higher nitrogen content results to a higher uptake from plants that will eventually result to a higher net primary productivity of a plant. As plant tissue senesces, nutrients are returned to soil as dead organic matter. However, before senescence occurs, plants absorb some nutrients from senescing parts to the perennial part of the plant. This process is called translocation or resorption.
Nitrogen cycle. Nitrogen is one of the most important constituents of all living organisms from bacteria to man. Animals obtain required amounts of nitrogen by eating plants and other animals. However, plants obtain nitrogen from either the soil or the air. Air has large amounts of nitrogen in its elemental form, but plants cannot use nitrogen in its elemental form. So, nitrogen should be converted to nitrogen-containing compounds such as ammonia, nitrites, and nitrates in order to be used by the plants. Some blue-green algae present in the bacteria can fix the atmospheric nitrogen into their protoplasm. These organisms convert the atmospheric nitrogen into ammonia. After converting this into other organic substances, plants utilize them for their growth. When animals eat these plants, these substances enter into the animal body. When these animals or plants are decomposed by the bacteria, nitrogen is released into the atmosphere. This is called the nitrogen cycle.
Nutrient cycling is achieved through the process of decomposition. Decomposition is the breakdown of chemical bonds formed during the concentration of plant and animal tissues. It involves the release of energy originally fixed by photosynthesis, carbon dioxide, and water, and ultimately the conversion of organic compounds into inorganic nutrients. It is a complex process of leaching, fragmentation, and change in physical and chemical structure, ingestion, and excretion of waste problems. Organisms involved in decomposition are categorized based on size and function. The four major groups are microflora, which is composed of bacteria, which is the dominant decomposer of dead animal matter, and fungi, which are decomposers of plant material. Both secrete enzymes for breakdown of complex organic compounds. A succession of microflora is involved in decomposition. The next group is mesofauna. That includes mites, potworms, and springtails. The next is macrofauna, and next is megafauna. Megafauna is composed of earthworms, millipedes, and snails. These decomposers obtain energy by consumption of organic material through oxidation in the process of respiration. Decomposition is usually studied using the litter bag's definition follows.
DFG Science TV, Function Through Diversity, pioneering work in China, the largest ever forest experiment on biodiversity.
The international team of researchers in China is primarily attempting to answer one key question. How does a forest ecosystem function and what role does biodiversity play in this? To answer this question, the researchers are studying the role of carbon and nitrogen in an ecosystem. They aim to track the routes these two elements take in the ecosystem. To achieve this, the scientists launch a large-scale litter decomposition experiment. Before they can begin, the researchers collect leaf litter using litter traps. Using these litter traps, they can calculate how much carbon and nitrogen from the trees and shrubs makes it back into the soil. The members of the team fill almost 1,000 litter bags containing leaves of one or more species. Then they spread them on the forest floor.
I'm doing the whole experiment because I'm interested in the carbon and nitrogen cycle, and the decomposition of organic material is a quite crucial process in the carbon-nitrogen cycling. And with all the data about litter fall, input, decomposition rate and so on, we can then calculate later the turnover rates of carbon, nitrogen and the cycling in this unique ecosystem here in Gubatan.
At regular intervals, they collect some of the litter bags, weigh them, and analyze their carbon and nitrogen content. The results of these measurements show which species have the highest rate of decomposition and whether the mixtures of litter from several species decompose faster. The doctoral researcher Yin-Lei Ma is also interested in the soil respiration. The question he's trying to answer is how much carbon dioxide is released by the soil to the atmosphere.
For this soil respiration measurement, I will start using this form. Yes, I have input the data, for example, the volume of the chamber and the area of the soil in the color into this form. Yes, and it will calculate automatically.
The researchers are also studying the forest soil to determine the amount of nutrients available for plant growth. In every soil layer, they measure the amount of nitrate and ammonium. These two nitrogen compounds play a crucial role in plant nutrition. Nitrogen is primarily needed for the enzymes involved in photosynthesis in the leaves. If there's a lack of nitrogen in the soil, plants grow more slowly. The researchers assume that forests with a high level of biodiversity are able to exploit the nutrients in the soil better. Thus, they are more productive than species-poor forests. The individual nutrients can be marked using chemical markers, making it possible to track how they're taken up by the plants. Over the coming months, the researchers will observe, on the basis of the nitrogen levels in this experiment, whether ecosystems with a higher level of biodiversity are able to exploit nutrients more efficiently. Back in the lab, the researchers are preparing the samples for analysis. They dry the plant matter and extract the nitrate and ammonium from the soil. In Peking and back in Europe, the prepared samples will then be analyzed to determine their carbon and nitrogen concentrations. With these findings, the team of researchers hopes to better understand the matter cycles in this species-rich ecosystem. Then they will also see what biodiversity contributes to these cycles.
Nitrogen is very essential for the survival of microbial and fungal organisms. For them to gather this nitrogen, these organisms must consume dead organic matter. In a process called mineralization, decomposers tend to transform organic compounds into inorganic compounds or mineral nutrients. At the same time, in a process called immobilization, these decomposers need to sustain themselves for their growth and reproduction by obtaining mineral nitrogen. Since these processes are occurring simultaneously during the decomposition of plant litter, the entire supply rate of mineral nutrients to the soil can be explained by net mineralization rate, or the difference between mineralization and immobilization rates. Changes in nitrogen contents of plant litter undergoes 3 stages. The first stage, leaching, involves the decrease of nitrogen content in the plant as water-soluble compounds are being leached from the litter. In the second stage, nitrogen content increases because of immobilization done by the decomposers. Finally, in the last stage, after getting sufficient amounts of nitrogen to sustain them, the nitrogen content of the plant will gradually decline. There is an altering of chemical compositions of residual organic matter in decomposition. Continuous degradation of plant litter will eventually make it become humus or dark-colored homogeneous organic matter. Soil organic matter is produced as humus embeds in a soil matrix. Decomposition of plant litter will continue until almost labile organic matter are consumed. Remnants of decomposition, referred as recalcitrant compounds, are increasing in the remaining litter since these compounds cannot be easily consumed by bacteria and fungi. Increasing residual organic matter will lead to the production of humus. Mineralized nitrogen are incorporated to this humus and will be combined with mineral particles to form solid colloids. Not all organic matter decomposes at the same rate. the rate of decay or mass loss is related to the quality of plant litter related to the types and quantities of carbon compounds present and features of the physical environment that directly influence decomposer populations, namely soil properties, that is texture and pH, and climate, which is temperature and precipitation.
During decomposition, microbes break through the leaf cuticle and traverse the interstitial space on their way to the cell wall. Microbes find food by releasing a small number of enzymes that break down complex compounds, like cellulose, into simpler ones, such as glucose, that can then be consumed. An increase in the concentration of these simple compounds tells the microbes to produce more of the needed enzymes for more release. The three main structural components of a leaf that microbes break down are cellulose, hemicellulose, and lignin. Microbes attack these high-energy foods, such as cellulose and hemicellulose, first. Lignin is a low-energy food, which shields cellulose and hemicellulose. Microbes only attack lignin as a last resort to get the protected high-energy food. There are two methods enzymes use to break down large, complex compounds into simpler, smaller ones that they can eat. One is the lock and key method, which is used to break down cellulose and hemicellulose molecules, which are chains of sugars, into single sugars that can be consumed. In this process, the enzyme is the key. The key fits the lock on the matching substrate molecule and causes a break in the chain. Some enzymes use this method to break a long chain in the middle to create shorter chains. Other enzymes clip off the ends to release individual sugars. Typically, multiple enzymes are packaged together into a complex scaffold. The scaffold organizes the enzymes that break bonds on both the ends and the center of the chain. To completely break down the chain, the scaffold slides up and down the chain and takes it apart. The other way enzymes break down complex compounds is by oxidation. This process is used to break down lignin. Lignin has a random structure. Every molecule is different, so the lock and key method does not work. Also, it takes a lot of energy to break down lignin, which is a very stable molecule. Because of this, enzymes do not have enough energy to attack lignin directly. Instead, enzymes attack other molecules that create free radicals, which are highly reactive compounds. These free radicals then react with the lignin and break it down. Once the lignin is removed in this way, the cellulose and hemicellulose that was encased by the lignin is available for decomposition. As microbes consume these foods, they are releasing carbon dioxide into the atmosphere through the process of respiration. Scientists need to understand how leaves decay and what influences how fast this process occurs so that they can predict how much carbon dioxide will be produced during decomposition.
Phytoplanktons have low lignin concentration and therefore decompose quickly, and some vascular plants can have lignin concentrations that are almost similar to those of terrestrials. Decomposition of plant litters is dependent on oxygen content of the water. Anaerobic bacteria carry out decomposition in mud and sediments of aquatic habitat. Absence of fungi, which need oxygen for respiration, hinders decomposition of lignin compounds and slows the overall rate of decomposition. Temperature and moisture greatly influence microbial activity. Lower temperatures or dry conditions reduce microbial activity, and decomposition rates are highest in warm, wet climates. Direct influence of temperature on decomposers result in a distinct diurnal pattern of microbial activity as measured by microbial respiration from the soil. Daily temperature pattern is closely paralleled by the release of carbon dioxide from the soil due to the respiration of microbial decomposers. Plant processes also enhance decomposition in the rhizosphere, which is a region of soil where plant roots function. It is also a place in the soil where decomposition is more efficient and faster compared to those in bulk soils. The roots play a very important role in decomposition as they alter the chemical composition of soil by secreting carbohydrates. Plant roots use carbon-rich exudates to supply the energy needed by bacteria for growth. As more bacteria is present, the decomposition process will be more rapid. The interplay between microbial decomposers and microbivores such as protozoa and nematodes determine the rate of nutrient cycling in the rhizosphere. Supplementing carbon to microbial decomposers enhances the decomposition in soil. The nutrient cycle doesn't only apply to those on terrestrial environments, it also applies to those in water. In coastal environments, the plant litter is submerged and there is faster decomposition than marsh due to the accessibility to detrivores and the presence of a stable physiological environment. In flowing water, fungi, leaves, twigs, and particulate matter are decomposed by different groups of shredders. In open waters, the particulate organic matter settles to the bottom and forms humic compounds. Lastly, in shallow waters, particles are fragmented and digested by bottom-dwelling detrivores. Basically, the material on the surface decomposes faster than the material that settles at the bottom. This is due to the different concentration of oxygen present. The decomposition of organic matter is the source of carbon for decomposition in aquatic environments. The major sources are macroalgae, phytoplanktons, and zooplanktons. Ciliates and zooplanktons digest the bacteria and excrete nutrients in the form of exudates and fecal matter. When the food is abundant, zooplanktons consume more than what they need and excrete half of that. An important component of aquatic decomposition is the microbial loop, where the decomposition of organic matter is introduced to the food web. The lighter materials stay at the surface for photosynthesis to support the net primary productivity. Nutrient cycling depends on primary production or inorganic to organic and decomposition which is organic to inorganic. Interaction of decomposition and primary productivity is limiting force in nutrient cycling or interdependence. For example, the cycling of nitrogen, which is an essential nutrient for plant growth. The maximum rate of photosynthesis is correlated with nitrogen concentration in leaves. Thus, the availability of nitrogen to the soil affects primary productivity via influence of nitrogen on photosynthesis and carbon uptake. Low nitrogen in the soil reduces net primary productivity, thus reducing both the total quantity of dead organic matter and nutrient concentration. Both the quantity and quality of organic matter influence the rates of decomposition and nitrogen mineralization or nutrient release. If it is high, it encourages high rates of net mineralization and nutrient supply in the soil. The changes in species composition and litter quality are directly correlated to the influence of soil texture on plant water available moistures. The water flow influences the nutrient cycling in streams and rivers. Inputs in the form of dead organic matter from adjacent terrestrial ecosystems, rainwater, and subsurface seepage bring nutrients into the streams. Internal cycling of nutrients follows the same general pathway as that for terrestrial and open water ecosystems. However, the continuous directional movement of water affects nutrient cycling in stream ecosystems. Nutrients are continuously being transported downstream, a spiral rather than a cycle, called nutrient spiraling. Nutrients in terrestrial and open water ecosystems are recycled more or less in place. An atom of nutrients passes from the soil or water column to plants and consumers and passes back to the soil or water in the form of dead organic matter. It is recycled in the same location within the ecosystem, although losses do occur. Cycling involves time. In water, a spatial cycle is added. Nutrients in the form of organic matter are constantly being carried downstream. Physical retention involves storage in wood detritus such as logs and snags, in debris caught in pools behind logs and boulders, in sediments, and in patches of aquatic vegetation. Biological retention occurs through the uptake and storage of nutrients in animal and plant tissue. The process of recycling, retention, and downstream transport may be pictured as a spiral lying horizontally in a stream. One cycle is the uptake of an atom of nutrient, its passage through the food chain, and its return to the water where it is available for reuse. Spiraling is measured as the distance needed to complete one cycle. The longer the distance of the spiral, the more open spiral. The shorter the distance of the spiral, the tighter the spiral. If dead leaves and other debris are physically held in place long enough to allow organisms to process the organic matter, the spiral is tight. This type of physical retention is especially important in fast headwater streams. Organisms that shed and fragment the organic matter can open the spiral by sending the organic matter downstream faster. Organisms that store dead organic matter tighten the spiral. Coastal ecosystems are among the most productive environments. As rivers meet the ocean, the current velocity drops and sediments are deposited within a short distance called a sediment trap. Buildup of sediments creates alluvial plains about the estuary, giving rise to mudflats and salt marshes that are dominated by grasses and small shrubs rooted in the mud and sediments. Nutrient cycling is different. It combines the features of each. Dominant plants are rooted in the sediments and function to link the zones of decomposition and primary production. Submerged plants take up nutrients from the sediments as well as directly from the water column. The directional or horizontal flow of waters transports organic matter and nutrients both into and out of the ecosystem. Nutrients are carried into the coastal marshes by precipitation, surface water, and groundwater. The rise and fall of water depth in the tidal cycle serves to flush out salts and other toxins from the marshes and brings in nutrients from the coastal waters by a process referred to as the tidal subsidy. The tidal cycle also serves to replace oxygen-depleted waters within the surface sediments with oxygenated water. The salt marsh is a detrital system with only a small portion of its primary production being consumed by herbivores. Almost 3/4 of detritus produced by the salt marsh ecosystem is broken down by bacteria and fungi. Nearly 50% of the total net primary productivity is lost through respiration by microbial decomposers. The low oxygen content of the sediments favors anaerobic bacteria, using sulfates rather than oxygen in the process of fermentation. A substantial portion of the NPP of a salt marsh is exported to adjacent estuaries. Each salt marsh differs in the way carbon and other nutrients move through the food web, as well as in the route taken and the amount of nutrients exported. Some salt marshes are dependent on tidal exchanges and import more than they export, whereas others export more than they import. Some material may be exported to the estuary in other ways. Primary production is regulated more by internal nutrient cycling than by external sources. Nutrients and oxygen are carried into the estuary by the tides. Typical estuaries maintain a salt wedge of intruding seawater on the bottom with a surface of flow of fresh water. The zone of maximum vertical difference in density, or the pinocline, functions much like the thermocline in lake systems. Living and dead particles settle through the pinocline into the countercurrent and are carried up the estuary along with their nutrient content, thus conserving the nutrients within the estuary rather than being flushed out to sea. The regular movement of fresh water and salt water into the estuary, coupled with the shallowness and turbulence, generally allows for sufficient vertical mixing. Surface ocean currents bring about vertical transport of nutrients. The global pattern of ocean surface currents influences patterns of surface water temperature, productivity, and nutrient cycling. The Coriolis effect drives the patterns of surface current. Lateral flow is limited to the upper 100 meters, but in certain regions, the lateral movement can bring about a vertical circulation or upwelling of water. Movement of surface waters offshore results in deeper, more nutrient-rich waters being transported vertically to the surface. Surface currents give rise to a similar pattern of upwelling in the equatorial waters. As the two equatorial currents flow west, they are deflected to the right, north of the equator, and to the left, south of the equator. When this occurs, subsurface water is transported vertically, bringing cold water rich in nutrients to the surface. These regions of nutrient-rich waters are highly productive and support some of the world's most important fisheries. Nutrient cycling is an essential process in all ecosystems and represents a direct or cyclic link between net primary productivity and decomposition. The link is variable, especially between terrestrial and aquatic ecosystems. There is a vertical separation between the zones of production and decomposition. In terrestrial ecosystems, plants bridge this physical separation. Roots provide access to nutrients available in the soil through decomposition, and the vascular system moves them to the sites of production. In aquatic ecosystems, plants do not always function as such. In shallow water environments, it is similar to those in terrestrial. As water depth increases, primary production is dominated by free-floating phytoplankton within the upper waters or the photic zone. In deeper waters, the zones of decomposition in the bottom sediments and waters or the benthic zone are physically separated from the surface waters where there is primary productivity. The physical separation is a major factor controlling the productivity of open water ecosystems. Vertical structure of open water ecosystems can be divided into three zones. The epilimnion or surface water is relatively warm and has a high oxygen content. It has high productivity and is poor in nutrients. The hypolimnion or deep water is cold and low in oxygen, which is nutrient-rich and low productivity. The transition zone is characterized by the thermocline layer. Warm, low-density surface layer of water on top of a cold layer of deep water is separated by a thin zone of thermocline. Winds that blow over water surface mix waters of epilimnion, but it does not extend into the hypolimnion. As autumn and winter approach, solar radiation decreases and surface water temperature declines, breaking down the thermocline and mixing the waters of the epilimnion and hypolimnion. It is called turnover, and nutrients from the bottom are brought to the surface in this process. With the onset of spring, increase in temperature and light in the epilimnion give rise to a peak in productivity due to the increase in availability of nutrients in the surface waters. The nutrient content will decrease as spring and summer progress, causing a decline in productivity.